Today we will deal with the topic of biotechnology. It is very important for the board examination point of view, and it is from NCERT Unit 9. This Unit 9 included two chapters, Chapter 11 Biotechnology, its principle and processes, and the Chapter 12 Biotechnology and its application. We will deal all these chapters step by step. And first, the Chapter 11 Biotechnology, its principle and process. This principle and process has been divided into some subtopics. First is its introduction, the historical background, principles, tools used in biotechnology, processes, and product of biotechnology. First, it just comes to the introduction. What is it? You will see whatever the study we do, or there is my simple question, why we study. Whatever the study is, this whole study, we can just say either it is philosophy or maths, whatever, science, art, whatever it is, all the human knowledge, all the human is always have a focus for the human welfare. It is very important that we always study just for us. This always focus on the human welfare. So we can say that the whole study, either it is philosophy, math, science, or anything, or understanding of natural phenomena, everything is anthropocentric. What it is? Anthropocentric. Anthropo, which means the human, and centric. It is anthropo, which deal with the human, and centric means focus. So the whole study is just anthropocentric. As we know, the human being is a most selfish creature of the planet. So now we come to the its examples. How the whole study is anthropocentric. First, we can just say when we study the physics, it will turn or give rise to engineering. Don't you see? If we study the chemistry, it will turn into industries. And if we study about biology, it is only focused on health and the food sources. Health and food sources. So it is proved now that the whole study of natural phenomena is anthropocentric. Okay. Then there is, as I am talking about biology, in the 20th century, biology has been turned into modern biology in form of biotechnology, which has improved a lot the health and the food product quality. It is very important. Improved the quality of health and food product. Okay. So we just see what is the history. We have seen the whole study is anthropocentric, and among them biology is mainly focused on the health and the food resources. And in the 20th century, which was just a change or offshoot of the biotechnology, in which the biotechnology has been turned into the modern biology in form of biotechnology. And by this biotechnology, it has qualitatively improved the food products and the health of human being. That's all. Now we have just covered this introduction. Now come to its historical background. There are mainly two names which had played a vital role in the foundation of biotechnology. First one was the Herbert Boyer, who was Herbert Boyer. What he has done, actually, he observed E. coli. He observed E. coli having an enzyme. This enzyme was restriction enzyme. What is this? Restriction enzyme. And this enzyme has capability. It is very important enzyme. This enzyme has a capability to cut DNA at a specific point. Okay, and after cutting, it left some sticky end. It left a sticky end. Why I am saying this sticky end? Because that cutted segment or the piece of segment after using the restriction enzyme was able to ligate or able to link with the another segment of DNA. So as we will see later that restriction enzyme is also known as molecular scissor. Why? Because it plays a very vital role in genetic engineering or biotechnology. Okay. Now, this specific, I had said the specific point. We will discuss it also later. What is that specific points where the restriction enzyme can be used? It was the first achievement. Now, come to the second one, who was Stanley Cohen. Second scientist who was Stanley Cohen. Stanley Cohen had developed a technique in that he observed the restriction enzyme. Here, the Stanley Cohen developed a technique where a plasmid developed a technique on study of plasmid that it can be removed. There was the first. It can be removed and reinserted into another cell. Okay, where it can multiply again. Let us see actually what is here going on. Stanley Cohen studied on the plasmid. What is this plasmid? Plasmid is the extra chromosomal part. What is this extra chromosomal part? Chromosomal part of either it is bacteria or any prokaryotic cell. Bacteria or any prokaryotic cell. So why it is going to be important? Because as it is extra chromosomal, but it multiplies itself because it is a part of DNA or it is a form of DNA. It multiply independently. It is very important point. So it is an extra chromosomal part of prokaryotic cell and multiply independently. Cohen had developed the technique in which the plasmid from the prokaryotic cell can be removed and this plasmid can be introduced into the another cell. Now, the technique of Robert Cohen and first as we have studied about Boyer's technique or Boyer's observation. What was his observation of restriction enzyme? The both has been combined and laid the foundation of biotechnology. Let us see how it has happened actually. So here it was Boyer and here it was Stanley Cohen. He developed restriction enzyme where with the help of this restriction enzyme we can just cut inserted in organism, host organism as we have discussed any another cell. Okay, this cutted piece of DNA actually what is going on is linked with the plasmid and form recombinant DNA. What it form? Recombinant DNA. And this recombinant DNA inserted into the host organism where this recombinant DNA will multiply and multiply or form clones, and in form of its multiplication we get product which is of our interest. Okay, it was the basic or we can say the basic principle of this whole biotechnology which was developed or founded by these both scientists Boyer and Cohen combinedly. Let us see exactly once again. There is a DNA. Okay, here this particular part, suppose this particular part is of our interest. This particular part is of our interest. We use restriction enzyme here, restriction enzyme, which will just cut it here and we can isolate or we get this desired DNA or desired genetic material. Then there is a plasmid which has been removed by any prokaryotic cell. This desired DNA and plasmid now combined and linked. Now it will form a recombinant DNA because the combination is new. So we call it recombinant. It is having our desired DNA and the plasmid. So we call it recombinant DNA. This recombinant DNA is now introduced in any cell. Suppose it is your E. coli. In the E. coli, it will just enter in this form. And as I have told, plasmid is nothing special. It is an extra chromosomal part of prokaryotic cell or E. coli which multiply autonomously. Now it will multiply autonomously. And as it multiply, it will form protein. As we know, DNA, then RNA, and then protein. So it will form protein in form of its product. So it has laid the foundation of biotechnology. So as we have seen the foundation, how it has been led by the two scientists Cohen and Boyer, we can define the biotechnology as it is a use of organisms and produce the product which is for human welfare. It is a technique. On the basis of this, the EFB, the European Federation Biotechnology has been defined. European Federation Biotechnology has defined this biotechnology as it is the integration of, we can just see integration of natural science and any living organism or its cell or any part of that organism or molecular analog. To get a result in form of product or service is biotechnology. Let us see again what is defined by biotechnology, or what is a biotechnology has been defined by EFB, which is European Federation Biotechnology. It is the integration or the combined study of natural science and organism or cells of any organism or any part of that organism, or we can just say molecular analog. And the whole study will be as a result of its product or service which will be helpful for the human. So it is also the human welfare main focused, and this whole is turned into the biotechnology. It was all about the introduction, historical background, and we can just say also that its definition. Now the next part of principle we will discuss in our next class, and that's all.